Hello, it's Mr. Nallis. Today we're going to be learning about cellular transport, and one of the things that we'll learn about today is actually osmosis. So here's a picture of Garfield trying to learn through osmosis. Now, I don't think it's working out exactly as he would want it to, but at the same time, at least he's trying. So before we go any further, I want to take a closer look and actually look at the cell membrane. And we're going to look at its structure. And we say a cell membrane has a fluid mosaic. It's kind of like a fluid mosaic model. So this is our current model for it. And it is a mosaic because it has, whoa, that was ugly. But it's a mosaic because it has all of these different parts and pieces to it. So here we have some proteins, and each and every one of these little orange circles or yellow circles, that's a phospholipid. And so it's a mosaic because of all the parts and pieces. And then finally, it's fluid because all of these parts and pieces are in flux. So they're constantly moving in and out and amongst each other. Now, while we look at this, there's one important thing that I want you to take note of, that this is made of a phospholipid bilayer. And this is basically a layer that has different charges to it, and it will let some things in the cell, and some things can't get through the cell, like water. So only small molecules like carbon dioxide and oxygen can travel and go kind of inside and out and move freely from a high to low concentration. But things like water actually have to go through something called a protein. So we have a specific protein for each molecule that needs to be let inside the cell. So a water molecule or something like glucose or a sugar can come here to the protein that's supposed to let it in, and it kind of has that lock key, and then it's going to unlock the protein, and the protein's going to let it inside the cell. And that's where our cells get things that they need, like sugar and water. Um, so there's a lot of different ways to get inside of the cell. But today we're going to learn about two different methods, and that is going to be passive transport and active transport. Now, last year we learned that all things move from a high concentration to a low concentration, and that's what passive transport is. It requires no energy because things naturally do that. So here's a little picture showing things, different molecules. Let's say this is oxygen going from a high concentration to a low concentration. And the cool thing about that is it requires no energy. So there it's going through the protein, and it's being basically let go inside or outside of the cell. Active transport is a little different. Active transport is actually going to require a little bit of ATP or energy, and it's going from a low concentration to a high concentration. So think about this. You have to spend a little bit of energy to bend over and pick up a dollar that's lying on the floor. But you know what? It's worth it. And your cell realizes that some molecules are worth it to spend a little bit of energy to bring inside of them. So that's more of an active transport, and it requires ATP. So um, we're going to talk about some of the different types of passive transports and active transports that your cell goes through. The first being diffusion. And diffusion is pretty simple. Basically what it is, it's just a molecule going from high to low concentration. And this is passive transport. It takes no energy to do this. So let's look at this picture. Let's say that this is um, dye, and I'm dropping a little bit of food coloring or a dye into water. Well, what's going to happen is it's going to start on this side, and all of these molecules are going to be in random motion. And eventually they will make it, and they will spread all the way throughout, and they're still moving in here, but it's at equilibrium, which is when there is an even distribution of the what we will call solute, or the sugar, or in our case, our food coloring molecules. Now, before we go any further, I do want to do a quick review over some vocab terms that you might need to know for this. And that is solute, solvent, and finally solution. So a solute is kind of like the sugar. And whatever it is, sugar, it could be glucose, sucrose, we put that into the solvent, which is generally always water. So water is the solvent of life, and then when the two combine, they become a solution. So in our previous example, the food coloring was our solute. Now, those three terms will be important for what we do with osmosis as well. And what I want you to know about osmosis, it is the diffusion of water through a semi-permeable membrane. Now, a semi-permeable membrane is basically a membrane where some things can pass through and other things cannot. So for our case today, our semi-permeable membrane, um, it can let water through, and all of these little orange or yellow particles, we'll say that's a glucose or a starch, all of these cannot pass through because they're too big. So water is going to move from a high concentration to a low concentration, just like diffusion. So it's the same thing, only it's the movement of water. So water here is our solvent, and the glucose or the starch, that sugar molecule, is our solute. Okay, but other than that, they're the same thing, and both of them are passive transport. So as you look at this, this is our little YouTube experiment. It kind of shows water moving from a high concentration to a low concentration. So in this example, we have more of a percentage of water over here. Let's say this is 70% water to 30% solute. And then it's going to go from 70% over to a lower concentration of water. And so that's the U-tube. It's actually going to shift over and the pressure is going to raise on this side. And here's another picture, and you can see that in this case the net flow of water is going to be from this side to going on the other side, because we have a higher concentration of water on this side, and the glucose, those are too big to get through our semi-permeable membrane. Now, when this happens, we can say that there are three different effects that water can have on cells, and these will be important. I want you to know these. The first is that we can have a hypertonic solution, and what this means is that it's going to be a high solute and a low water concentration. Okay, so that's a hypertonic solution. A hypotonic solution is when we have a low solute and a high water concentration. Finally, isotonic solution is when the water concentration and the solute, they are equal inside and outside of the cell. So with this, where does this actually take place? Well, osmosis constantly takes place within our red blood cells. And when we have a hypertonic solution, that means we have, hyper means like it's a lot of sugar. So we have a lot of sugar in our solution. And what's going to happen is water is actually going to travel from high concentration to low concentration. In this case, it's going to travel from the inside of the cell to the actual outside of the cell. And what's going to happen is the cell will shrivel. Okay, now when we have an isotonic solution, we have water going inside and outside of the cell at the same rate. And this is at equilibrium right now. So this is kind of what we want. That's an isotonic solution. Same amount of concentration inside and outside the cell. Finally, when we have a hypotonic solution, what takes place is we have more of a water concentration on the outside of the cell than inside of the cell. So water is going to travel to the inside of the cell, and what can take place is the cell can actually burst because of this. And this is the cool part about plant cells, is they have this extra barrier. That's the cell wall around there. And the cell wall keeps it rigid, and that's how it protects the plant cells from actually bursting, when this can actually take place within like a red blood cell. So those three terms will be important. So take another look at this and see if you can kind of figure out the differences between each. So that's osmosis, and that wraps up passive transport. So diffusion, osmosis, those are passive transport, require no energy. Active transport is going to be a little different. So with active transport, one way this can happen, and the picture shows this, is that we can have something, and like I said before, we have specific proteins that require energy, but it's going to let this particle inside of the cell. And it requires energy. That's the main thing to know about active transport. So it can happen like this. Active transport can also happen, um, and this is kind of cool. It's called endocytosis, and with this, it's a process of taking large material into the cell by the infolding of the membrane. So if you look at this picture, there's this little spot right here where we have an infolding of all of these different molecules. Let's say that this is a virus, and the cell is actually inviting this virus in because it recognizes it, and what's going to happen, it's going to hook up to a lysosome. The lysosome will release its digestive enzyme and destroy all of those viruses. So your cell does a massive transport, and it takes large material into the cell by doing this. Um, and it lets more things in at a quicker rate. The other way was through up here where we just have a protein, and that's another different kind of active transport. So that's endocytosis. And the cell also does something called exocytosis. And this is almost the exact opposite, but this is when we have a bunch of waste built up in the cell, and it's going to go to the cell membrane. It's going to kind of fuse together here, and then it's going to release all of those different parts. It could be a waste, it could just be something it wants to release into the body as well. So that's exocytosis, and that also requires a little bit of energy as well. Now, that kind of wraps up the difference between active and passive transport. So today we learned about diffusion, osmosis, semi-permeable membranes, isotonic, hypertonic, and hypotonic solution. And that seems like a lot, but hopefully you kind of got the grasp of how all of those different parts work within the cell and how a cell membrane actually functions as well. And finally, hopefully you can connect diffusion or osmosis with something that we see in our daily lives. Okay, think about this. All of you have experienced diffusion before, because I'm most certainly sure that you have walked into dog poop, came up into the classroom, and then students around you first notice that, oh, something smells kind of funny, and then finally it fills up the entire class. That's diffusion. So try to connect it to something that you know. And then finally, you should be able to look at a cell and look at the concentrations of water inside and outside the cell and determine, is it hypertonic, hypotonic, or isotonic. So that's the cell membrane, that's cell transport, and I hope this was helpful.